Hello everyone, this is Uzma Afreen from Birla Institute of Technology, Mishra, India. Today I am going to talk about a prospective approach to combat wheat leaf rust disease with the help of microRNA. Sample provided by ICAR, IARI and funded by UGC Government of India. My aim is to develop a novel strategy to protect wheat seedlings against leaf rust infection. My objectives are listed here. Putting some back information, leaf rust is caused by fungus Puccinia triticina. It reduces wheat productivity severely. As said by Norman Borlaug, you can't build peace on empty stomachs. The concern intensifies after seeing the hike in hunger ratio, mostly in developing countries like India. This is a rust tracker map of year 2020. The red circle is showing the high severity of rust disease in India and its neighboring countries. So, how to combat this? Here is a catch: regulatory microRNA. MicroRNA are small non-coding RNAs that regulate gene expression with the help of RNA interference mechanism and show greater role in biotic and abiotic stresses in plant. MicroRNA like RNA are same as microRNAs, but their origin are different. So for this, we have constructed four small RNA libraries from leaves of wheat near isogenic lines of susceptible and resistant wheat plants, inoculated with uridinous spores of Puccinia and mock inoculated. These small RNA library are mapped with each other to get unmapped reads. From these unmapped reads, novel microRNA and microRNA like RNA identified following a proper bioinformatic pipeline. Their target genes are predicted, validated, annotated, and their differential expression analysis was performed with the help of quantitative real-time PCR. In table 1 out of 100% of susceptible small RNA reads, 14.29% of reads were unmapped. In table 2 out of 100% of reads, 39.29% of reads were unmapped. Figure 1 is a Venn diagram showing total predicted novel microRNAs among the four libraries. Table 3 is the selected novel microRNAs out of the total predicted microRNA. They are divided into three categories, that is resistant novel microRNA, susceptible novel microRNA, and susceptible resistant common novel microRNA. In table 4, the predicted wheat target genes of exclusive resistant novel microRNAs are listed with their descriptive function. In table 5, the predicted wheat and Puccinia targets of resistant susceptible common novel microRNAs are there with their descriptive functions. The highlighted row in both the columns are explained further. These are the target gene ontology analysis graph plots showing that wheat plants do respond towards stress. These are the target T plots generated by hitting the degradome library. The red arrow indicates the cleavage site on target gene. The long sequence containing the red and blue sequence is the precursor microRNA sequence, which gets folded into a stable secondary hairpin structure. The red and the blue sequence are excised out with the help of protein machinery. The red sequence is known as antisense microRNA and the blue sequence is known as mature microRNA sequence, which mature microRNA sequence do complementary seed pairing with its target gene. Here we can see that one microRNA is target pairing with two genes. This is a flowchart showing the expression status of all novel microRNA and their respective target genes. In figure 11, the expression of microRNA 8 is directly proportional to the expression of its target, that is leaf rust resistant protein in figure 12. As the infection time point increases, the expression of microRNA 8 increases. So if we increase more the expression of microRNA 8 artificially in wheat plants, the wheat plants will be resistant towards leaf rust. In figure 13, the expression of microRNA C is inversely proportional to the expression of its target syntaxin 16 in figure 14. Syntaxin 16 in Puccinia plays role in membrane trafficking. So if we increase the expression of microRNA C artificially in wheat plant, there will be a communication gap between host and pathogen interaction. From the total resistant genes, Majority of the genes are located in chromosome number 1, which could be further used in wheat breeding programs. Our microRNA of interest are encapsulated in the nanoparticles which will be sprayed on the wheat seedling to keep it safe from leaf rust. My conclusions are one common microRNA indicate the possibility of co-functional microRNA, resistance susceptible and pathogenesis related novel microRNA like RNA and microRNA are found in wheat and Puccinia. Puccinia microRNA like RNA indicates a strong cross-kingdom communication. MicroRNA expressed differentially illustrate that microRNA expression is activated upon Puccinia attack. One microRNA with multiple targets. Functional annotation of target genes reveal that microRNA enhances plant resistance. Most of the selected disease-resistant genes are located on wheat chromosome number 1, which could be further used for the management of leaf rust disease. Thank you.